Boa tarde a todos, dando continuidade ao terceiro dia, né, do nosso quarto SInPeM. Eu gostaria de agradecer imensamente a todos que tornaram a realização desse evento possível, especialmente para as pessoas que estão me auxiliando aqui nessa conferência na parte da tarde, né, ao nosso aluno, né, o Leonardo, nossa egressa Joselaine e também ao Kleberson, que tem atuado, né, na parte técnica, né, permitindo que esse evento transcorra tão tranquilamente online. E nós teremos, né, o apoio do Alcélio e da Rayane na tradução em Libras, e teremos o Sérgio atuando como tradutor aqui para facilitar a comunicação com o professor Freek Bakker, a quem eu agradeço imensamente por aceitar o nosso convite de participar, compartilhar com a gente algumas ideias muito interessantes que ele publicou recentemente num artigo com coautores, né? O professor Freek Bakker, ele está na Universidade de Wageningen, na Holanda, e ele é um biólogo, trabalha com sistemática, estudos comparados, né, ele tem um foco especial agora, nesse momento, pelo que eu entendi, na extração de DNA antigo, né, de exemplares de herbário, que isso é uma tendência muito grande nos museus de história natural, tanto para herbário quanto para animais, poder utilizar as novas ferramentas que nós temos, né, para acessar essa informação que está nesses grandes museus, né, com essas novas tecnologias, mas o artigo que foi publicado, né, recentemente, ele trata de uma questão muito interessante que diz respeito a esse processo de digitalização e disponibilização de dados, dos metadados dessas imensas coleções reunidas em diversos museus e de algumas oportunidades e desafios, né, que isso tem proporcionado agora no século XXI. Então, eu agradeço muito ao professor Freek por compartilhar essa experiência conosco e passo a palavra a ele.
Ok, obrigado. Let me quickly, yes, this should work. So, hello everybody. I hope you can see my first slide here, okay?
Yes.
So, great. So first of all, thanks for inviting me here to your very nice symposium. It's a pleasure to be here, and it's also a nice challenge for me to be in a somewhat different community that I'm usually in, and I'm looking forward to that. So what I would like to do today with you is indeed to share some thoughts on the idea of the global museum. And first of all, as Isabel already introduced, I'm a biologist, and I am not in a museum. So I'm in a university, but I have had some collaborations and ongoing work with museums. So there is definitely the interest there. Now, just to see where I'm from, this is the Netherlands, and you see in the background Denmark. Here is Amsterdam, Utrecht, and here is Wageningen, which is actually quite a small town in the east of the country. And this is our campus. You know, it's all life sciences and agricultural sciences, and hardly any humanities or languages. So it's pretty much a life science university. And yeah, as a biologist, I've been studying systematics and phylogenetics of different groups, but most of all the genus Pelargonium, which is seen over here, which is a member of the Geraniaceae family. And this is sort of the kind of biologist that I am, doing comparative biology and systematics of a group of species. Now, Pelargonium, you may recognize the flowers perhaps, and also there is of course geranium and the others. But Pelargonium is quite amazing because it is a group of 280 species that has quite a lot of variation in terms of floral morphology, but also in terms of leaf morphology and overall shape. So there is lots of evolution packed here in these almost 300 species that enables us to test hypotheses on how things change at an evolutionary time frame. Here are some setups in the greenhouse that we can use to do crosses. This is the work of a PhD student of mine 2 years ago, and here just half a year ago, to also look at the genetics underlying how species can or cannot cross. In all our work, collections are pretty important. There is obviously the idea of the tree of life, which is sort of the evolutionary tree of everything, ultimately based on collections. And in that pattern of the tree of life, there's also process involved, and we aim to understand both process after the patterns have been elucidated. And this entails different disciplines. There's phylogeny reconstruction, there is biogeography, global change science, there is taxonomy, phylogeography, evo-devo, the evolutionary development of things, speciation as a process. And then outside these fields, you can think of sort of an applied fringe where we have things like pest control, leaning on speciation knowledge, plant breeding on evo-devo, DNA barcode based on taxonomy, etc., etc. This is what I tell my students, that this is the systematics arena, and that it is quite interesting to be in this arena, especially where we have like a biodiversity crisis. We have taxonomic impediments, meaning not enough taxonomists anymore to describe the numerous amount of species still out there. We have a next generation technology where there's lots of good opportunities there to do big data science. And bioinformatics is becoming more and more important there. So systematics. A couple of years ago, I was active in the so-called Consortium for the Barcode of Life. And this generated my interest further into the use of collections. Because what we were after in this consortium is to try and get the DNA barcoder in place, which would be having a little machine, putting a bit of DNA or tissue of any specimens that you want to identify, put it in, and hopefully after a few minutes, the name and the information would appear on the screen. Now, this machine is not yet there, but in principle, the technology and the databases are all there to do that. And then later on, I was lucky enough to be able to coordinate one of the joint research activities in the synthesis. Is a European Union funded big taxonomic facility program coordinated from London. And what I could do there is to focus on these questions having to do with herbarium DNA. And can we use collections to get DNA out and do all sorts of things? So questions like, oh, that would be nice, but do we then optimize our extraction protocols? Should we worry about post-mortem damage? It's one thing to get DNA sequences out of museum specimens, but is the data reliable? Can we sacrifice bits of old precious type specimens? Is it worth doing that? And how can we know that? can we do genomics on herbarium DNA? And what about contamination and other questions? So this yielded a bunch of papers and some activities that was quite nice to do. And based on these papers, yeah, you know, I was invited to all these interesting places to come and tell the story, how to do this. And that indicated that there was indeed quite some interest, I think, in this field. And indeed, I still see that increasing today, how to exploit collections for DNA. During that project, we did lots of things, and one of them was indeed looking at DNA extraction, and there are a couple of factors there, the specimen age. Do older specimens yield worse DNA or not? What about plant secondary chemistry? The compounds, the chemical compounds present in plant leaves. And what about tissue preservation? What do we know how the specimens were fixed when they were collected, after they were collected? And these will all contribute to the efficiency of herbarium DNA extraction. So we did lots of studies there, and this is Martijn Staats, my postdoc at the time. And Martijn, he looked at a couple of trees that were growing in the Leiden botanical gardens in the Netherlands, and for which old herbarium had been made. So in a way, it's a bit like this. Here is 1900, here is 2000, here is now. Here are the trees that were already growing in the botanical gardens. This is Martijn, my postdoc. And he got old herbarium that was made in the 1900s and tried to get the DNA out. And of course, the trees are still there, the same trees. And so this enabled us to look at modern specimens and modern DNA taken from the same trees. So that way you can compare old and fresh DNA. Now that yielded several conclusions, but basically the message was it's surprisingly well preserved in our herbarium specimens. And this is the so-called dot plot, where you can see the chloroplast genome. This is like 150,000 nucleotides in DNA compared with itself. And this is a specimen of 146 years old. And almost the entire genome is reconstructed, no problem. So this was pretty good news and promising to use collections in more intense ways. And we published some bioinformatics pipeline to do that and try to spread the message. And this, I think, also led to me being invited to this particular workshop then in 2017, and it was called The Role of Museums in Modern Evolutionary Biology. And what happened there is that Professor Scott Edwards from Harvard University, from the Zoological Museum at Harvard, he had been given a grant to organize these topical workshops in Sweden, in Gothenburg. So this was quite nice. We all went to Gothenburg, and this was the workshop for a couple of days. And of course, we had lots of good times as well, and discussing the wonderful opportunities and possibilities out there. And basically, the people involved were studying a wide range of topics, all related to collections, of course. Insights from tracking evolutionary novelty, quite an interesting study, building of infrastructures for investigations of a changing planet. This actually also included how to use mammalian tissues to study virus evolution, and how topical this has become, of course, in the recent years. And it turned out that these zoological museums are increasingly important to find and track old and new viruses and how things are developing, because the viruses are in the specimens, no problem. You can just sequence them and get them out. Some more case studies where collections turned out to be really important. Communities, island communities, invasive species, biological invasions, being able to look back in time and use collections to reconstruct what actually happens. my work on herbarium DNA, the new technologies like 3D imaging of huge specimens, very interesting how that can be used in combination sometimes with CT scanning on smaller specimens. Citizen science as an important contribution to how our collections are growing and being maintained. And then natural history museums as research infrastructures, and lots of it was also focused on ornithology collections where many things are happening. So the whole workshop then led to this paper that we were able to publish in Peer J, and it has received a decent amount of views and reads and cited in 33 articles, I believe, right now. And it's nice to see how this is taking on. And when we were in the workshop, we were discussing the different missions and different ideas that might be important in order to assess the role of museum collections in modern evolutionary biology. So here is a word cloud of the notes and the paper that we made. And we were saying things like this: natural history museums harbor the authoritative records of biological diversity across time and have always been meeting places for scientists, amateurs, and the like. That lay citizens tacitly endorse the information given at museums in a time when many institutions are bitterly distrusted. So the special role for museums and natural history museums to just believe what you see. So our paper was then sort of pitched at, let's say, a more general scientific public and also to policy makers. And then global museum, well, yeah, if you Google it, you get these sort of things also. And it was quite interesting to see where this term is being used and what it represents. For instance, there is, of course, sort of a platform for museum news, views, and vacancies and what's going on in the museum world. Maybe you will be familiar with that. Discussions on where do we go from here, and some critical notes on museums in the US. So this was another workshop several years ago, apparently, and I understood that there was some discussion there, especially in the humanities and the arts, and that the Global Museum has drawn quite some attention. Let me just try and get this. That's better. Quite some attention, and issues there turned out to be mostly what is good taste. Interesting question. Correcting post-colonial perceptions. That's appears to be quite a big issue. Deprovincializing, for instance, by buying the Guggenheim license in order to become global and become a global museum and benefit from increased visitor numbers. And there is also a global museum in the San Francisco State Museum Studies, a space in which to appreciate the power of diverse communities in a globally connected world, as they phrase it themselves. Global Museum has also been considered a shorthand for a debate on the different approaches that museums are or should be taking. And as always, quite interesting there is how we can combine or use the regional perspective versus a global perspective. In any case, try to raise political consciousness of the audience of museums. And in the US, I found some discussion on whether museums are undergoing an identity crisis, that they would have invested lots of capital for long times in order to create a globally oriented identities and operations in their museums, and that they now must reckon with the results of maybe sometimes flawed decisions and would risk the development of a more isolationism feeling in society, and this happened especially a bit in the Trump administration. So there was some concern there. And then what was this global museum? Was it a branding ploy to justify ambitious and aggressive expansions? And if so, then yeah, the global museum would no longer be simply a warehouse for antiques, but that there would be a mission, of course, through staff diversification, public programming initiatives, that it would raise its audience political consciousness. And many in the art world would hope that the Global Museum would become a tool of cultural diplomacy, a bastion for multiculturalism, or at least a guardian against Philistinism. According to this critique in this newspaper issue in 2017. So I found that quite interesting to see that these kinds of discussions are going on. In the natural history world, things are maybe a bit different. What we consider to be a global museum there is not so much a debate, but the potential global community of natural history museum collections that increasingly are becoming interconnected through digitization and thereby enabling both synergy and increased power of resolution in testing, mostly, I put it here, evolutionary or historical biological hypotheses. So our view would be that in the Global Museum, scientists worldwide would be able to study and sample each other's specimens, including from remote collections, perform geometric morphometric analyses of shapes, or generate high-resolution CT scanning data and compare related images. So there would be both a national and a regional focus in natural history collections, and regionality, yeah, is considered a strength because it could help local communities to be sort of proud at their own flora or fauna or their biological setting. And not being global could actually mean, yeah, a strength in that sense. Coming from colonialist enterprises, especially in Europe, international specimen acquisition study is now conducted usually in strong partnership with the local institutions, fortunately. What we see is that citizen science is contributing an increasingly part, in spite of an increasingly urban population, perhaps surprisingly. We've seen large initiatives such as GGBN, the Global Genomic Biodiversity Network, DISCO in Europe, which was the follow-up of the synthesis program, trying to connect European natural history museum collections, and IDIC-Bio in the US, doing more or less the same thing, but slightly different setup. So museums, therefore, pretty much in focus, and despite their immense value, yeah, it's no news to you, I guess, that museums facing grand challenges. In terms of natural history, we can say that there are challenges in terms of taxonomic impediment, meaning there's not enough taxonomists curating certain groups. So the groups may become neglected somewhat in the collection. Of course, there's a funding challenge. Regulated species administrative burdens may prevent further development of collections. What about the coordination of secure data standards? There have been quite some efforts there to try and reach that, but is it actually working? Yeah, and then of course, more physically, the distribution, the risk spreading, the redundancy in the digitization of collections, and unfortunately, the natural heritage loss. And yeah, these examples may probably be very well known to you, especially your National Museum in Rio de Janeiro. That was such a dramatic aspect, as well as the Butantan Museum here in Sao Paulo. Of course, that were terrible losses to the science. And maybe it's interesting to compare that legacy lost to what happened in 2017 in Paris, where the Notre Dame Cathedral, sort of the central symbol almost of Catholic churches, caught fire. And it's interesting to see that, well, I think within a month or so, there were millions and millions and millions of euros gathered and raised to repair and to fix this damage. And I'm not sure if that also happened like with the National Museum in Rio de Janeiro in the same speed. Now, I just throw this in. This is quite an interesting document written by the National Academies of Science and Genetic Medicine in the US on the value and prospects for biological collections. There's actually a nice, the whole book you can download here for free. And I found it quite an interesting overview of the challenges and possibilities in there. Now, back then to natural history collections, and indeed the global initiatives that have been developed over the decades. One of them, probably the most famous one, is Gbift, the Global Biodiversity Information Facility. And this is basically a huge data set. At the moment, I think, well, this 1.3 billion records that would comprise both observations and specimens. And at the moment, I understand that it's more like 80% is observations rather than specimens in there. And there are some issues with the reliability of identifications. But anyway, it's still a very important central facility and really important to be able to use that. I'm just checking here on the chat. Is everything okay in terms of, can you still hear and see me okay?
Yes, everything okay. You can see you and.
Okay, good. Yeah. So that was Gbift, and other impressive global initiatives are, for instance, the Planetary Biodiversity Mission, with this kind of numbers and budgets aimed to map or identify as many specimens as possible in local areas, and combining all these existing huge institutions or platforms together. So in biodiversity science, people are, finally, are getting the act together, and the community is, I think, making quite some good progress. Here is just a list of what we call the aggregators of biodiversity data. We published this in the PRJ paper. And this is basically a list of what institutions and databases are there. And indeed, it's spreading quite a bit, including databases with phylogenetic trees and taxonomies and all that, distributed either in the US or more in Europe or truly globally. Now, and then there is citizen science, which indeed we see is increasing quite a bit in importance, both for maybe collecting data and collecting specimens and feeding it to the museum collections, or maybe helping out in the collections for identifying specimens. We see that also a lot, which can be extremely useful. And then there is also the databases here like iNaturalist, where photos can be uploaded, pictures dated and with lat-long locality information that produces an extremely valuable and growing database, which can be used to test certain hypotheses, especially related to global change biology. Now, the global museum then would be, or can be envisaged to have all these different stakeholders. And in this figure, I put in yellow the flow of data, and in green, the flow of specimens. So a main stakeholder would, of course, be science in general, like these areas: conservation biology, ecology, emerging pathogens is of course rather topical, and evolutionary biology. That would include these factors to study. And we see that with science there is a flow both ways in terms of data and in terms of specimens. For other things, there may be just only data flow and no specimens. And then there are other sample providers that would indeed feed the collections with more specimens and get data back in return. And there is other ways of doing the educational outreach, for instance, getting data out and specimens out to be used in educational activities. Now, the biological specimen then, I think it's useful spending some minutes on that. It obviously is a voucher, not only of the actual individual sampled at the time, but also often of its locality, including information about the soil, about other biotic and abiotic conditions in which the specimen was collected. Where was it collected, and what was the edaphics, the soil situation, temperature, the altitude? All these metadata come with the specimen. Inside specimens, we have microfloras, bacterial communities. There is a lot to be gained from a biological specimen. Specimens would allow repeatability, sampling things again. So repeatability both in terms of precision, but also accuracy. What kind of trend would you like to track or monitor, and does it work okay? Specimens could be considered to be realized genotypes, or maybe, genetic variation, and some variants made it and others didn't, and the ones that made it are the specimens that we collect. So specimen collections, therefore, perhaps as large phenotyping facilities, test and proven through the millions of years to be working at that specific genotype. Of course, the time series that can be built, specimens collected in 2 years' time from now, can be compared with those 200 years ago. It's obvious, but if you think of it, it's quite a good opportunity. The discussion about specimens versus mere observations, obviously specimens are much more valuable, you could argue. Only sometimes observations can replace specimens, for instance, when ethical, conservational, or practical issues are concerned. Then, of course, we must allow to have observations instead. And there could be observation biases of several kinds, indeed. This will probably differ per group that's been studied. Now, here is a nice example, I think, of what we can do with biological specimens. I call this previous pathogens. You may know about the Irish Great Famine in the 1840s that killed so many people, starvation, and it was caused by Phytophthora, a little creature it's related to diatoms and brown algae. And it swept through the potato crops that were used at that moment. And fortunately, there was some herbarium made of those potato plants at the time. And from there, Phytophthora mitochondrial genomes, like 15,000 bases, could be sequenced and were put in a phylogenetic tree. And it turned out all these isolates were what we call monophyletic. They stem from one most recent common ancestor. So that means that this horrible thing happened just once and hasn't been seen again, fortunately. And this is valuable information for the epidemiologists studying these outbreaks, and especially where it came from and how it crossed the world. Another example, ancient alleles. This turns out to be a specimen that was dated in the, I think, 18th century, and it appeared to have a certain substitution in a gene that's associated with herbicide resistance. But it's interesting because that mutation, that substitution, was already there before herbicides were invented and used. So why is it there, and against what other force could these have developed? We only can know this because having the historical specimen in place. There is the idea of the extended specimen, therefore, which would add value to our specimen collections. Collections that would indeed comprise, obviously, locality info, tissue, the tissue itself, the DNA itself, the biopolymers, isotopes, metabolites are in there, and then high-resolution CT scanning data and SEM, electron microscopy images, is all adding tremendously to the specimen value. But all this new data needs new storage. For instance, image data is known to use a lot of space for properly storing it. Here are some examples of some structures that are part of the alligator tracheae, the breathing airways, compared to alligators and ducks. And this has been the result of a CT scanning experiment, and storing that so that you can look from all sides inside the specimen also takes quite some space. And this is an example from a study, Kingsley et al. In PNAS, where they say this is an integrated approach incorporating data and perspectives from different disciplines, from developmental biology, from focal physiology, from paleontology, and it defines a promising agenda for understanding the evolution of the sea rings. And I'll show you in a second what that means. So integration of all these different data streams will allow bridging among disciplines and involvement of engineering, biomedical sciences, and perhaps also art. So this is what this particular study focused on. Here we see a phylogenetic tree of the vertebrates, let's say vertebrates starting with your amphibians, and they call it tetrapods starting here, indeed. Amniote, reptiles, Archosaurus, extinct lineages, and birds over here. And this is ourselves, tetrapoda here. And plotted on this phylogenetic tree are the probably homologous structures in the larynx. And the larynx is present in most lineages, also in ourselves, but in different extensions, and has developed in different ways. Now, if you look closer to the branching of the tracheates over here, this is all just normal branching, with the tubes either being fused or slightly separated, or fused or separated again, or fused. And then here we see in birds that this part has yielded a new structure, which is called the syrinx. And that structure is involved in vocalization. Now, that's all very interesting, but the deeper question here is that the larynx is also still there, and the larynx also produces sound. So in evolutionary biology, it's then very interesting to think of how can we have a new structure arising with an overlapping function with what was already there. So how can we understand that new functions come about when the old functions are still doing fine? This would not be feasible within a Darwinian framework, when it's all about natural selection being the survival of the fittest, and then new structures could only arise if there would be tremendous new fitness added. But that's not clear in this case. So an interesting fundamental evolutionary question. And thanks to the availability of specimens in museum collections, the authors did a lot of different kinds of studies. So here is that question again formulated. And why would a novel structure evolve to maintain the same function? From spandrel to novelty, well, spandrels is something that's used quite a bit in evolutionary biology. It refers to the spandrels of San Marcos, the famous church in Venice. And this was coined by Stephen Jay Gould, the famous and popular books writing evolutionary biologist at Harvard. Spandrels are the bits, I understand, between the ribs that make the ceiling of a cathedral. So these bits in between, they are just there. They have to be there, but they don't seem to have a specific function. And this is then compared with what we sometimes see in the evolution of living things. Sometimes things are just there without an apparent function, and that could be this split of the tracheae, where we have something sitting there, and this could have evolved in now what adding these focal flaps there in the syrinx structure. If that's the case, you want to know about the function of this thing. Is there stress on the wall, shearing? So you probably want to do some modeling. And in any case, there will be different data streams combined. Using many museum specimens, I spoke with the co-authors of this paper, and they went to every natural history collection in the world and tried to compare all these particularly homologous structures and try to use that in their modeling studies. So here is some of the modeling that's being done to actually find out which places of the structure would have a lot of stress and would have low stress, and how that would perhaps then explain why we see the cartilage rings occurring in those structures. And maybe that actually using Turing models, the models that were developed by Alan Turing, a mathematician in the UK, might explain how these rings are separated, maybe even better than trying to find homology among all these bits and pieces. And then what about the evolution of this larynx and this new structure, the syrinx? Maybe they use the same gene networks, maybe not. There are all sorts of interesting new evolutionary hypotheses can be formulated from such studies, including in their sort of overall conclusions that during the evolution of all these lineages, you would have your larynx, the old bone structure producing vocalization, the syrinx not yet being there. There would have been some sort of functional overlap time with a quiet zone where there would be perhaps the larynx not producing sounds and the new structure not yet. And then ultimately the syrinx now would produce vocalization and having taken over from the old function. Now that's an interesting case to be able to study. And again, we need the collection specimens to check and do the simulations and provide supporting evidence for such hypotheses. Other examples, maybe movements, skeletal movements, the way forces and movements are occurring, and how you can mimic that. This is also referred to as biomimetics, and this could be quite important for developing the next generations of robots. So collections then, perhaps better seen as incubators of innovation. Specimen collections enabling answers to a large number of other scientific questions, some of which have not yet been posed. We don't know yet what questions will be answerable. Increasingly, museums are leveraging new data from their specimens, and integration of data types allows training in techniques that bridge among the disciplines. So collection-derived data sets that are of relevance to disparate traditional fields such as engineering, biomedical sciences, or arts. Collection-derived data sets may actually be seen to bridge those areas. So therefore, the natural history museums would increasingly serve as a nexus for work that disregards disciplinary boundaries and addresses questions we didn't know to ask before. For instance, this syrinx example, how could it evolve when the old function was still there? So collections may actually inspire anti-disciplinary approaches, appreciating the importance of the specimen in any case, natural history collections as incubators of innovations. And this is just one way of visualizing that. Perhaps in your museums, in the natural history museums, you would have your exhibition part, your collection part. People tend to get inspired by the exhibitions, become educated about it. Obviously, it all evolves around specimens. More interest will probably lead to people going into the collections, get more information, study their work, and have some sort of innovation that could then feed back into the exhibition for new inspiration circles, and all happening within the Natural History Museum. Now, for all of this to work properly, we would need to have continued and comprehensive collecting. As mentioned, in these time series, specimens collected 200 years time ago can be compared with those collected in 200 years or the other way around. And this is valuable because it will allow hypothesis testing on community change, for instance, due to human influence. There are some very nice examples. We also discussed that in our paper, like mollusk surveys over, well, almost hundreds of years, and see how composition, but also morphology of individual species will have changed in a correlation with environmental change. There's been some good publications about that. But there's also idiosyncrasies and biases in natural history collections, because many will have grown through research programs of individual scientists. It's just a given fact, I think. Frequently, rarities have been emphasized, which causes quite a bias. And in general, there could be quite patchy collecting habits going on, not systematically collecting. And especially evolutionary biology would actually benefit from better common species representation in collections worldwide. Your house sparrow or other globally occurring common species are usually not well represented in collections. If they would, evolutionary biologists would much better be able to test evolutionary hypotheses on these lineages. Only having rarities is not always very useful. Yeah, and then there are other aspects, like ethical aspects. So this is a story you may have come across, I believe, a couple of years ago. An ornithologist, Christopher Filardi, found a bird. He had searched more than 2 decades for it, and then collected it. So this was in the South Pacific, in some little islands. And yeah, finally he had rediscovered it, and there was like 2,000 individuals on that island, so a decent sort of population. And he collected it as is normal in taxonomy, which means you kill one individual. And after he had done that, he was quite excited about it and happy to let people know via social media, I believe, that he had found this thing, which caused an enormous uproar. That, yeah, people were really upset about this beautiful bird, and how could he have killed one individual, you know? And this really turned very ugly. The poor man had to hide, and his family was chased by people standing in his garden, and horrible story, but it was true. And so the conclusion can be drawn that in this case, we see that the loss of life incurred by collection of a single bird caused a lot of emotional response, much more rather than a scientific appraisal of the true impact of collecting data on populations, which is far less. And besides, what about, you know, damage by like feral predators such as house cats? They cause a lot of damage and far less emotional response. Do we hear about that? Something on place-based discovery. It may seem obvious, but still, when the collecting efforts from the perspective of different clades have focused on a particular location, there is an additional layer of comparative information. Species correlations, environmental correlates, edaphic correlates, and the evolution of communities, the full suite of local biodiversity. Now, taking place into account enables asking different kinds of questions and can lead to place-based discovery. And Miller puts it like this: places are not simply a semantic convenience, no, they are a meaningful lens for viewing the world, because it's orderly with respect to geographic space. So for biological collections, the place-based approach is relevant as it seeks to understand how local processes connect with those at larger scale, like global scale perhaps even. So superimposing different kinds of layers, indeed, from like edaphics, chemistry, geology, archaeology, geography, may give therefore a holistic specimen, in a sense comparable to species distribution modeling using maxent. I don't know if you're familiar with that, but in biodiversity science, this is used a lot to try and predict where a particular species may be able to occur, given its sort of ecological envelope and giving baseline data on a certain area. So place-based approaches would enable better understanding and even prediction of change in biological systems over time. And here are some examples. This is a very nice South African Cape example, taking into account all these interactions and then coming to different sort of predictions. And it can give serendipity of collecting data over periods of change identified later, perhaps, as well as interaction among scientists sharing an interest in the same geographical location. And I know some very nice studies where anthropologists, botanists, and zoologists together studied certain areas and came to very interesting additional ideas and hypotheses to be testing. And here is another example of such a place-based discovery. It's the Moreira Island in the South Pacific. And this is a so-called digital ecosystem avatar. The project's called Moreira Idea. Moreira has been the subject of DNA barcoding like a decade ago, that every single bit of life on the island was attempted to DNA barcode to have a nice register of what's out there on the island. But in this case, it is a simulation platform to try and test and visualize different scenarios of what might happen, combined with real baseline data of the islands. So you can do all sorts of different aspects that you may want to focus on and see how this would turn out in different scenarios. Now, as said earlier, from a natural history perspective, the Global Museum would entail the potential global community of natural history museum collections that increasingly are becoming interconnected through digitization, enabling synergy and increased power of resolution in testing mostly evolutionary or historical biological hypotheses. So scientists worldwide would be able to study and sample each other's specimens, including from remote collections. This is to some extent already going on. There is also remote microscopy programs that you can actually drive a microscope on the other end of the world in another collection. Genomic resources, the GGBN network, for instance, excuse me, perform like geometric morphometric analyses of shapes across specimens, or generate high resolution CT data and compare related images. In any case, the Global Museum can do more than individual museums can, by linking data from various kinds and by offering increased taxonomic resolution, maybe a bit not unlike a synthesis radio telescope. Like here, we have 16 radio telescopes. The signal is combined and then processed, and together this gives an enormous added power that single telescopes would never have. So think of each telescope representing one natural history collection. Or for instance, here the other way around, signals are processed and then combined, may also be one way of doing it. Or here, where telescopes may actually be movable or not fixed in a position and can be sort of adjusted to whatever resolution of power is needed for that particular question. And if you would think that of natural history collections, maybe in the future things may be reorganized, or for certain clades or lineages, we may see different constellations of collections being grouped together. I hope this. There we are. So, and this could then hopefully yield what's been named by these authors as to replace parachute science with global science, to have the broader perspective, but in the same time still being anchored in the regional perspective as well. Now, finally, then, a few examples of the Global Museum generating interesting results. Here is a study a few years ago already, integrating natural history collections and comparative genomics to study the genetic architecture of convergent evolution. And this meant that these people would go into the collections and try to find examples of convergent evolution, getting the same structure from different unrelated lineages, and then looking at the genomics behind that. And they suggest that the synergistic and complementary roles of natural history collection derived phenomic data and comparative genomics methods can be particularly powerful in together elucidating the genomic basis of convergent evolution among higher taxa. Genomic studies of convergence would be greatly improved by integrating deep vertical natural history knowledge with horizontal knowledge focusing on the breadth of taxonomic diversity. So in this comparative genomics, there is still a lot to be gained from exploiting these collections. Natural history collections have and continue to be best positioned for increasing our comprehensive understanding of phenotypic diversity, is their conclusion, with modern practices of digitization and databasing of morphological traits, providing exciting improvements in our ability to evaluate the degree of morphological convergence. Mind you, this has been attempted to do based on like model systems where you have just a few lineages where you understand to some extent how things are organized, but not having the horizontal breadth as it's mentioned here. A last example, DNA barcoding, an example of integrating collections worldwide. DNA barcoding, well, you may be familiar with that, maybe not, but in any case, it's the idea to have a standardized region of the genome, that's database, so that when you have an unknown sample of a specimen, you can sequence it and compare it against the database and know the identity, species identity of your organism. And all the data are put together in Bolt, Barcode of Life database, and you can just go there and get yourself a free account and do your searches, or see what is known about your favorite group of species. For instance, for my Geraniaceae, my Pelargoniums, there is 296 published records distributed like that. And here you can then see what groups, what labs are actually contributing to this particular project. So anybody who would be interested to contribute could now phone up these people and say, hey, I may have some specimens as well, and maybe that will fit very nicely in the sampling that the other groups have, and filling gaps that were left over in the sampling of other institutions. So together integrating this in this sort of virtual workbench of BOLT is, I think, a very nice example how comparative biology is getting forward, producing then this sort of updated species counts and how each species is now covered by different specimen samplings. So, okay, and sometimes I also got the question that, okay, this is nice, your global museum, but how will it work and what is needed in order to expedite things further? You could perhaps say that indeed for a synergistic global museum to happen across a wide spectrum, coordination, infrastructure, governance structure, and data standards are needed. Data ownership, visibility, transparency could perhaps be improved using emerging blockchain technologies, like the history of a specimen throughout collecting, identification, changed opinions, maybe shifted to another part of the collection, renamed, whatever. DNA is being extracted. Blockchain technologies would be a very secure way of tracking all those histories. And then, of course, it's needed to sell the idea of the global museum, a formidable challenge. And how will policymakers as well as the general public be convinced of the importance of a global museum as a top priority? Maybe naming it evolutionary science instead of biology might help. And here's an example of the so-called overwhelmingly large telescope, which was a follow-up from the Large Telescope project and the Super Large Telescope project. And this is what the physics community has done so very well. They came up with the overwhelmingly large telescope program and were able to sell it to get the funding and make it happen. So, and then I come to promoting science through museums indeed. In order to enhance the broader relevance of natural history museums, it is indeed important to message effectively to industry, to policymakers, to bridge the traditional chasms between science, the arts, and the humanities. And why not use specimens to do this, to make the bridge and to find new ways of using them? Because in a way, you could argue that both arts and science depend on inspiration, creativity, and critical assessment. And this is what museums, specimens usually offer. And there are some examples, like in the Porto Museum in Portugal, the Hall of Biodiversity, where this kind of beautiful displays have been produced, or like this, that could indeed help for this goal to make people fascinated about specimens in a different way. Now, the relevance of museums should also be increased through digitization and imaging, obviously. It's important to do that, but also the names, proper updates of names, nomenclature, make sure that that's in order. Digitization, of course, is a huge effort. Should we digitize blindly, going through everything, or is there some priority that needs to be put in place? Obviously, as mentioned, maybe tracking specimen history by blockchain technology, combining specimen databases, resolving redundancies to see what is where in excess or in overlapping numbers. Yeah, for big clades, large collections, for instance, in Leiden, in Naturalis, I know there are huge collections of certain groups of butterflies. Digitization may become quite challenging, and is there enough infrastructure? So usually the big clades are sort of left for last. BOLT, the DNA barcoding database, and also UNITE, the fungal database, are very good examples of a reference framework for aiding in specimen identifications. Yeah, the reference collections that are needed. So in conclusion then, the natural history museums, indeed, for hundreds of years have around the world provided the general public and scientists with numerous opportunities to learn more about the natural world. Taking together this global museum must be seen as one of the most valuable assets of modern society and culture. This is actually what we state in our paper. So therefore, natural history museums as academic nexus of integration can function as an interdisciplinary meeting places or innovation incubators where questions are addressed that we didn't consider asking before. So I think quite exciting prospect. The core of these institutions are the specimens. So we need to think about how to store, preserve, handle, and do things to them. In the view of maybe indeed new unforeseen need. I mean, especially in biopolymers, we don't know what we'll be able to do like in protein chemistry in the next 10 or 15 years. So it's better to store it already in a way that could be useful in next generations. Viewing natural history museums as critical infrastructure for scientific inquiry and public understanding may help raise their profile and awareness, facilitating continued support. We hope so. And with that, ladies and gentlemen, I'd like to thank you for your attention. And yeah, let's hear any questions or discussions that you may want to start on these. So thank you.
Thank you, Freek, for this enormous quantity of information for us.
Yeah, it was a bit much perhaps, but yeah. How are we doing in time?
It's exciting, very exciting to think about natural history museums in the 21st century. You are just showing. Desculpa, eu posso falar em português, né? Não é isso. eu vou até falar em português, que eu acho que facilita, né? Então, é muito interessante.
That's good, yeah.
Muito interessante. Se você tiver qualquer dificuldade em ouvir a tradução, por favor, me informe, tá? E aí eu posso tentar falar em inglês para a gente não perder a comunicação, ok?
No, I can hear the interpreter very well. Yeah, okay. It's good.
Então, assim, com essa enorme quantidade de dados sobre esse aumento da relevância dos museus de história natural e das suas coleções no século XXI, onde a gente tem novas questões que já se colocaram e novas que ainda podem se colocar em relação a problemas que são centrais para a humanidade, né? Aqui você deu exemplos de questão de saúde pública, eu acho que barcoding tem uma direta aplicação na questão da segurança alimentar e muitos desdobramentos que são aplicados, inclusive, né? Que a gente começa a se tornar relevante, as nossas coleções começam, e os nossos dados, metadados, passam a ser relevantes para outras áreas de conhecimento, né? Eu acho isso muito importante, porque eu vejo um grande paradoxo, que justamente me parece que os museus de história natural chegam ao século XXI muito mal financiados. Eu acho que esse não é um problema, as coleções carecem enormemente de financiamento público, seja na Europa, nas Américas, onde for, né? E a gente vê que esse desinvestimento que vai na forma da não contratação de especialistas para estudar essas coleções, porque nós estamos falando de frentes novas, muito excitantes, mas os museus de história natural ainda lidam com os problemas tradicionais, principalmente em áreas de mega diversidade, como é o Brasil, de lidar com questões básicas de inventário da biodiversidade, né? E, nesse sentido, eu vejo que os museus enfrentam dificuldades imensas, e isso me parece um grande paradoxo. Depois da Convenção da Diversidade Biológica, o Protocolo de Nagoya, os países... os países assumindo compromissos, né, na geração de dados. Eu, por exemplo, me preocupo quando o investimento público vai muito mais na digitalização, para uma disponibilização rápida dos dados, do que na salvaguarda dos acervos, e nós vemos incêndios enormes destruindo a base da biodiversidade in situ e ex situ. Então, assim, como lidar com esse paradoxo onde a gente tem coisas extremamente excitantes acontecendo, projetando para o futuro, e carregamos ainda problemas pesados e fundamentais, porque sem as coleções, sem a base material para os futuros estudos, tudo isso não irá se realizar. A gente está condenando as futuras gerações a um apagão de informação que enfim, insuperável.
Now that's indeed a paradox. It's quite concerning, absolutely. And I think this idea of selling the idea and making policymakers and politicians aware of the huge possibilities and opportunities. I mean, take this corona pandemics and how collections are so valuable to trace what's actually been going on in hosts, in animal hosts, and that has to be done to make people aware of the importance of these collections. I was involved in the DNA barcoding movement 15 years ago, and also there we hope that instead of calling something taxonomy, now we call it DNA barcoding, which actually is doing the same thing in a way, but it sounded much better, and there have been cases where funding was much more easy to get. It's a bit tricking the system perhaps, but it did turn out to be successful, and especially the group in Canada, in Guelph, they have been able to raise millions and millions of dollars to get this idea forward, and it all came down to, in the end, the curation of specimens, because a DNA barcode is only a DNA barcode if there is a specimen. That's the whole idea. So it trickled down into traditional taxonomic research. So it is a matter of telling the story, and our paper was sort of meant to be pitched to those people, and hopefully they will read it. But
eu tenho só uma outra provocação que eu queria fazer, te perguntar, porque você mencionou que talvez se substituir o nome de biologia por ciências evolutivas, né?
Evolutionary science.
Você acha que é mais apelativo? Eu lhe pergunto se, na verdade, se a gente tratasse como ciências ambientais, que eu acho que é um tema que hoje a gente consegue articular, talvez, preocupações e atenções mais globais. Eu não sei se... eu acho que ciências evolutivas em partes do planeta talvez fechasse mais portas do que a própria biologia, né? Assim, com essa questão que a gente ainda tem negacionismo científico e tudo mais.
Okay, well, that's interesting indeed. You mean perhaps because of sensitive issues in communities that don't like evolution for religious reasons. Yeah, you're absolutely right. That's true. Yeah, we have to take that into account. Yeah.
É apenas uma provocação, assim. Temos perguntas para o professor Freek?
Nós temos uma, e eu vou fazer aqui, uma pergunta que veio anônima, mas está falando nesse sentido. Vou utilizar o inglês também para a gente se aproximar um pouquinho mais aqui. A pergunta é a seguinte: Thinking about a place for research, do you believe that the Global Museum needs to be linked to a physical space, or can surveys be carried out just in online environment?
Sorry, could you repeat the question? I didn't get the first bit.
Okay. Thinking about a place for research, do you believe that the Global Museums needs to be linked to a physical space, or can surveys be carried out just in online environment?
I think the online environment will be good enough. Will be already contributing and opening up possibilities. If we have increasingly faster internet and good technology there, like sharing the CT scan data and being able to compare specimens across the world quickly, I think there is a lot of promise there. What can be done? The 3D scans, especially, also to link up things physically. Probably with especially with having meetings and exchange of ideas, that should perhaps be more happening even more. There is, of course, the whole notion of carbon dioxide footprints and not traveling so much, and that is true. That is definitely even more so the opportunity to try and do it online. But things like sharing genomic extracts, that's already well underway just by posts and by the GGBN, and that's also a huge opportunity to fill gaps in the sampling of different data sets reciprocally. So indeed, sometimes people do ask that, will there be a huge building somewhere that's called Global Museum? Probably not, but...
Eu penso que nenhum museu gostaria de abrir mão, né, de ceder suas coleções para um museu global. Isso não aconteceria de forma alguma, enquanto a ideia de compartilhar os dados certamente é muito bem-vinda, né? Mas eu vejo só uma última provocação, desculpa, professor, mas você pode depois falar, é que eu vejo, na verdade, pelo menos no futuro próximo, assim, ou imediatamente, eu vejo essa tendência da construção desse museu global, que é uma coisa, a digitalização da informação taxonômica, ela vem já de muito tempo, ela é tradicional, a gente tem a digitalização de publicações, de dados, de coleções. Eu fiz meu mestrado na década de 90 e eu podia procurar os espécimes e os museus, onde estavam alocados os espécimes que eu trabalharia e estudaria, em bases de dados que já estavam disponíveis. E eu vejo, na verdade, isso como um grande instrumento de pesquisa agora. Vejo menos a relação com educação e outreach do que com a pesquisa. Eu vejo como um grande instrumento de pesquisa. E queria ouvir você falando sobre essa questão.
Well, in itself, that is a good thing, but like I showed in this diagram, with the exhibitions and the collections, exhibitions are a vital part of the whole museum endeavor, of course, I would like to think. So how to then translate the new innovations and the exciting new insights into the exhibitions and let the general public be enjoying that and be inspired by that. And as I said in the beginning of my presentation, I believe that there is this notion that many institutes are not trusted anymore by the general public, like science in general, or the church, or hospitals, or we have these amazing discussions going on in Europe. But usually natural history museums are still trustworthy by most people. They will believe what it is said on the labels, and no reason to doubt all that. Maybe apart from strong creationists, but but I think as long as the exhibition side and the general public side is maintained, it will be fine to grow larger research collections, because you don't want to end up in a situation where some populist politician may take over government and decide, you guys in your ivory tower, what you're doing, I don't understand, and I don't want to have it anymore. I don't know whether this answers your question to some extent, but...
Na verdade, eu entendo, mas é porque, como as exposições são tradicionais, dos museus tradicionais, assim, eu pensei como que a gente teria essa expansão de ordem, de grandeza, na potencialidade de outreach com um museu global, mas eu acho que precisamos estudar os dados, olhar os dados e experimentar a potencialidade desses dados da forma como eles estarão configurados, né? E que usos o público poderá fazer, né? Porque tem tantas peculiaridades. Você mencionou a questão da confiabilidade das identificações. Isso é uma coisa que preocupa muito, né, os museus de história natural, porque nós, especialistas, sabemos que até quão confiável é aquele dado que foi publicado, mas não sabemos o uso que será feito por um usuário não especialista desta informação, e essa é uma preocupação muito grande, né?
Is this especially for biological collections, or also like cultural or humanitarian?
Eu penso em coleções biológicas, eu estou falando aqui, pensando nas coleções biológicas.
And you mean that there could be like an intellectual property or a safety aspect on how data may become used?
Exatamente, para o público, assim, projetos educativos com essa informação, como na verdade, digerir, triar e transformar isso em alguma coisa que faça sentido para o público não especialista.
Yeah, that is indeed an important mission. That's true. But that would then be the exhibition side, I guess, how that is. And maybe also, like what I mentioned, to try and bridge the gaps with art, and having, art as an effective messenger of what things are about. Like the example in Porto Museum. Maybe that could also be enhancing. Sometimes natural history museums have this sort of rather dusty appearance, or dimly lit, and it smells funny, or and that may not be the best way to.
Pode ser, alguns sim, alguns não todos, mas enfim, temos tempo para mais uma pergunta?
No, indeed,
Eu acho que nós temos mais uma questão, e aí também a gente já atinge o nosso horário, acredito que podemos fazer essa. Então a questão é a seguinte, professor.
Okay.
Quando falamos em Museu Global, existem consensos ou protocolos internacionais quanto a quais metadados decidir publicar em bancos de dados internacionais? Poderia falar um pouco sobre esse processo de decisões informacionais e protocolos internacionais, se houver, por favor? É o Cleberson Moura que envia para a gente.
Okay. Well, thanks for that question. That's indeed an. Let me just, by the way, quickly plug in my power because I see my battery is going a bit. 1 second.
Eu ia falar para ele, Isabel, quando ele voltar, o tradutor pode traduzir, que o Museu de Zoologia aqui de São Paulo não tem, assim, não está coberto de poeira, né? Nem cheiro esquisito.
No, but maybe I can answer that. Very good. That's, yeah, sorry about that generalization perhaps, but very good, yeah. So, but with regards to question on the types of data and protocols, yeah, I've seen a little bit about that within the European large projects such as Synthesis, and there are these working groups that work on standards of data. There is now, of course, also the Nagoya aspect that everything needs to be, yeah, accountable and in terms of the origin of the specimen. And this is slightly different matter actually, but I did not mention it in my presentation, but this could be one of the main obstacles to the use of global museum, because the Nagoya legislation could ask from us to be able to assess what legislation was at the time of collecting in that particular country, and whether using the specimen now is violating that legislation or not. And this could be a huge bureaucratic burden on the use of specimens, and it's not completely clear yet, as I understand. But then the question about what kind of data, I think it's pretty much agreed upon, like your obvious metadata, you know, like altitude or soil types, or yeah, and there are also these standards. I think the TEDWIC working group has meetings every 2 years and try to update the standards and what should be recorded and what not and how. And I would like to think that this is connected with the US digitization project, IDIG-Bio. I don't know what's in Brazil, what kind of digitization projects are on the go, but most likely they will all communicate about the data standards and what to import. I don't know of any issues where really people did not want to see their data being shared with others, or no, so far I haven't. So.
Bom, eu acho que é isso, né? Então, eu agradeço muitíssimo a Freek por compartilhar com a gente essas ideias, assim, apresentar esse cenário que é extremamente...
De nada.
Dos museus de história natural, né? E agradeço a todos que colaboraram aqui para a realização dessa conferência e informo que estamos partindo para as salas das apresentações dos trabalhos, tá? Nós começamos agora às 4 horas, às 16 horas, a apresentação dos trabalhos nas diversas salas, tá? Então, ótima apresentação para os que vão expor seus trabalhos. Professor Freek, mais uma vez, muito obrigada.
It was a pleasure being here. It's really, yeah, I'm glad to, thanks for the invitation also, yeah.
Thank you.
Okay, and best of luck with the meeting. Yeah, I was very busy with teaching, so I couldn't follow many things, but, well, it's...
Obrigado, professor. Eu só queria também me apresentar para colocar uma carinha no e-mail que você recebeu, né, Joselaine? Obrigado novamente pela disposição, né, e pela apresentação de hoje.
Yes, of course. Yeah.
Me too.
It was a
Esperamos
pleasure.
vê-lo no Brasil.
Thank you.
Esperamos vê-lo no Brasil, quem sabe? São Paulo.
That will be very nice. Absolutely. Yes, let's go for that. Yeah. Great.
I also would like to thank you for being here with us today. It was such a great moment. And I hope you see your next opportunities too, over here in Brazil or all over the world. We are talking about a global museum, so where can it be? We don't know.
That's right. We should be everywhere. Absolutely. No, but thanks. Yeah. Okay. Yeah. Okay then.
Boa tarde a todos. Encerramos, então, mais essa conferência.